Hello, everyone. My name is Brian Shi, and I'm a senior in the MSCC program at Paul Laurence Dunbar High School. Today, I am proud to be presenting my 360-hour capstone research project, The Effect of the Ketogenic Diet on Cognition Relating to Alzheimer's Disease. Before I begin, I'd just like to thank my mentor, Dr. Eileen Lin, for giving me the opportunity to work in her lab to conduct this research. Now, I want to give a quick overview of what I'll be covering today. First, I'd like to give you all some background on my research. Next, I'll present my research question and hypothesis. I'll continue into explaining my methodology and segue straight into my data. Afterwards, I'll make some final conclusions, examine future directions, and thank all those who helped me along the way. Lastly, I'll display all of my references and take any questions. So let's get right into the background. Alzheimer's disease is an extremely serious and common neurological disorder that causes brain cells to degenerate, leading to deteriorated mental functions, including worsened memory and impaired thinking/reasoning. It affects 5.8 million Americans, and it typically affects older people. The amyloid hypothesis that is most commonly believed states that the accumulation of amyloid beta clumping together to form deposits in the brain leads to the neurodegeneration that's commonly seen in Alzheimer's. The picture on this slide shows amyloid beta clumping together to form a plaque, which is a hallmark of Alzheimer's disease. In other words, the amyloid hypothesis theorizes that amyloid beta is the main root of Alzheimer's. And with this in mind, a lot of time and a lot of money was spent on developing drugs with amyloid beta as the therapeutic target. However, these drugs have pretty much all ended in failure, and as of right now, there's no effective therapy or treatment for Alzheimer's disease. And so, therefore, my project is focused on the neuroenergetic hypothesis, which runs contrary to the amyloid hypothesis. That is, this theory believes that Alzheimer's disease is the product of energy deficiency in the brain, as glucose, the brain's primary source of energy, becomes increasingly less able to cross the blood-brain barrier and supply the brain with energy. The stress that this energy deficiency causes is hypothesized to lead to many of the symptoms of Alzheimer's disease, including amyloid beta deposition. With a ketogenic diet, a diet high in fat and low in carbohydrates, glucose is replaced as the primary source of energy. Protein pretty much stays the same, and so it's just considered at moderate levels. And so the aim of the ketogenic diet is to cause controlled ketosis in the body, so instead of burning carbohydrates, the body breaks down fats. This leads to the production of ketone bodies as fuel. Thus, ketone bodies could potentially be an alternative source of energy and could even improve brain function. You see, ketone bodies are one of the few other molecules that could bypass the blood-brain barrier other than glucose. And the depicted picture on this slide is a reference for some of the typical foods of a ketogenic diet for humans, including fish and including avocados and including cheese. However, in this research project, this wasn't exactly the types of food that were fed to our test subjects, but I'll explain a little bit more on that later. So there are 3 alleles for the ApoE gene that feature in my project. The ApoE gene is found on the 19th chromosome, with ApoE2 being protective against Alzheimer's and it being the least common allele. ApoE3 has no impact on Alzheimer's disease and is the most common allele. ApoE4 increases the risk of getting Alzheimer's disease significantly, and it is slightly more common than ApoE2. So the differences in these 3 alleles are just different amino acids at 2 sites, yet the amount that these 2 different amino acids affect the risk of getting Alzheimer's disease is positively huge. So moving on, my investigative question is, could the ketogenic diet potentially act to prevent the onset of Alzheimer's disease? I'll be examining the effects of the ketogenic diet, and I hypothesize that it will improve cognition. For my methodology, my lab ordered 3-month-old transgenic ApoE3 and ApoE4 mice from the National Institute of Health. They were then fed for 16 weeks with either the ketogenic or control diets. The ketogenic diet resembled a white paste, and it would be put in the mice's cages in a cup. The control diet was just standard food pellets. The mice were fed ad libitum, which basically means they could eat as much of the diet as they wanted. And so after the 16 weeks of feeding, it was time for behavioral testing for cognition and motor activity. But I'll explain a little bit more about how these behavioral tests work on my next slide. MR spectroscopy was done on the mice to examine their metabolite levels in the brain, specifically glutamate. Finally, data analysis using the software PRISM was conducted to determine significance. So figure 4 depicts the open field test, which was used to measure motor activity. Mice were allowed to roam around for 5 minutes in an open field, as the name suggests. There are 4 open fields in the figure with a mouse in each. We did it like this so that it would take less time to get through all of the mice. And then EthoVision software was implemented to track the mice to see the distance traveled. And figure 5 depicts the novel object recognition test used to measure memory. On the first day, there are 2 objects that look exactly the same, and the mice become habituated with it. On the second day, one of the objects is replaced with a new object that these mice haven't seen before. If the mice spend more time exploring the novel object rather than the object they were already habituated with, then they are considered to have better memory. The EthoVision software was again used in the novel object recognition test because it could track where the mouse's nose was and where the mouse's tail was. If the mouse's nose was faced towards the object, then it would be considered to be exploring the object. And so the software counted the amount of time that it spent exploring the novel object and the amount of time that the mouse took to explore the old, already familiar object. And by comparing the two, it was able to get the data that we wanted. Figure 6 depicts a mouse being prepared for MR spectroscopy, which is basically using the same machine as an MRI scan. Now moving on to my findings, the ApoE4 genotype of mice performed significantly worse on the novel object recognition test. And this makes sense because the ApoE4 genotype of mice are significantly more at risk for Alzheimer's and likely have some cognitive impairments already. Meanwhile, figure 8 shows that the mice on the ketogenic diet perform significantly better on the NOR test, potentially showing improved memory. The mice on the ketogenic diet also have significantly higher glutamate concentrations in the brain as well. Given that glutamate concentrations are decreased in Alzheimer's disease, the ketogenic diet could potentially bring glutamate levels back to normal. Glutamate plays an important role in memory, and its reduction is involved in the pathophysiology of Alzheimer's. Glutamate is also a precursor for GABA, which plays an important role in learning and in supporting muscle function. Moreover, glutamate is yet another energy source that can compensate for the decreased glucose metabolism that's linked to Alzheimer's. Figure 10 conveys that the mice on the ketogenic diet move significantly more on the open field test. This could potentially mean that the mice are more energetic due to the ketone bodies. Another possibility is that the ketogenic diet led to an increase in motor ability, which is supported by existing literature stating that the ketogenic diet may potentially improve neuromuscular function. In conclusion, my hypothesis was supported. The ketogenic diet improved memory irrespective of genotype. The data that involved glutamate and distance traveled was also very promising, as it may help support the neuroenergetic hypothesis, and it portrays other beneficial effects of the ketogenic diet. The glutamate findings and the distance traveled findings could also potentially be linked with the improved memory findings of the novel object recognition test, as glutamate is linked to memory, and the increase in distance traveled could potentially be linked to an increase in energy supply to the brain. However, one limitation that my project does have is that my sample size was a little small, and so it was difficult to achieve significant figures. Thus, I had to combine the ApoE3 and ApoE4 genotype of the mice in order to achieve significance. However, we can still see that irrespective of genotype, the memory, glutamate concentrations, and distance traveled for these mice were all still improved. And as for my future directions, I'd like to examine the effects of the ketogenic diet on the gut microbiome and cerebral blood flow. The coronavirus epidemic prevented me from getting cerebral blood flow results for Senior Symposium, but these are definitely effects that I would like to investigate in the future. I'd also like to look into other nutritional approaches to Alzheimer's disease, as I really didn't believe in the beneficial effects of nutrition in combating disease until I started conducting this research. Finally, I would want to explore human studies of the ketogenic diet to see if these beneficial effects can be translated from mice to humans. I now like to thank everyone who's helped me along the way. Dr. Lin helped guide me through this entire research process as my mentor and has taught me so much about the scientific process. Andrew helped train me, and the other members of the Lin Lab were extremely supportive and a joy to be around. I'd also like to thank the University of Kentucky, Mrs. Young, and the Kentucky Young Researchers Program for granting me the opportunity to conduct research as a high school student. The Math, Science, and Technology Center has taught me so much and been an amazing environment to spend 4 of my most formative years. Finally, I'd like to thank my family for putting up with me and being supportive, and my friends for always giving me rides to lab. I really have appreciated everyone that's helped me along the way. And so here are my references. And if you have any questions, please email me at bmshi77@gmail.com. Thanks for listening. Bye.